UV light, it just glows. It is like
we are ready for our
like
the
almost
second
parts per
of the
trillion.
double feature for
No,
this
billion, sorry.
afternoon. I want to thank, we have a brand new set of people, so I want to thank them all for coming and remind you all that there are opportunities to watch this again on YouTube. So if you go to YouTube and put in Chemistry, Lawrence, and Senior, you can pull all of them up and watch them. And so that it's my distinct pleasure to introduce Chelsea. Chelsea worked with me this summer, along with Casonia Smith, on a project that I've been working on for many a year, and that they helped put the last few nails in the coffin of a very nice paper, which they are authors on. But they also, as you'll find out from Chelsea's work, opened a whole new can of worms as to all kinds of questions that we can ask. And it's been a distinct pleasure to have Chelsea around. It's sad that she's graduating. Well, but Casonia will be around for one more year, so that will count. And that Chelsea is going to be taking a year off, and then heading off to pharmacy school is the plan. So anyhow, without further ado, Chelsea.
All right, well, thanks for that awesome introduction by Dave. I don't know how I can follow that up. So yeah, I'm Chelsea. The topic of my talk is the asthma-related gene matrix metallopeptidase 12, or MMP12, and how it's regulated by human rhinovirus exposure in macrophages. So quite the mouthful. Yeah, I know, I see you non-science people being like, asthma what? So we're going to start with asthma. Most people know what it is. It affects somewhere between 6 to 8% of all adults in the United States and 8 to 10% of children. So it's a crazy amount of people that actually have asthma. Chances are most of you know somebody who has asthma, and just judging by the number of people in this room, maybe 4 of you have it, maybe? I don't know, somewhere around there. Anyway, it's a breathing disorder that is the result of an inflammatory constriction in your airways. So if you look at this picture, this airway is a normal, non-asthmatic airway. It has a thin airway wall and a thin layer of muscle surrounding it with a giant hole for air to easily flow in and out of. And next to it on the right, you see the same airway during an asthma attack. So there's a thicker wall, the muscles have contracted and are much thicker and causing it to be aggravated. And there's even a thin layer of mucus in there. So that just leaves a tiny little slit for air to actually get in and out of your lungs. So asthma occurs in these life-threatening attacks, generally characterized by difficulty breathing, obviously, tiny slit, wheezing as a result of trying to force air through a much narrower airway, a red face, just miserable people in general. There are lots of different triggers that asthmatics have. It can be something like pollen, dust, cats, dogs, even exercise if you are unfortunate enough to have exercise-induced asthma. So really the main problem with asthma is this inflammation that's occurring in your airways. So inflammation is just the body's immune response to the presence of non-self that is there. So any pathogens, it's really just working to try to get that out. But the problem with asthma is that it's actually causing the airway to be remodeled, and the airway thickens. So it's actually not so good for the asthmatics. Okay, so yeah, so when I'm saying remodeling, what I mean is that there's just an extra collagen production from your epithelial cells. So this collagen is permanently narrowing your airways, so every time you have an asthma attack, your airways are going to get slightly more and more narrowed just because you can't break down all that extra collagen that's being made. And this collagen production is coming from inflammatory cells that are releasing inflammatory mediators. Inflammatory mediators would be things like cytokines, which are proteins that are used in cell signaling, proteases, which are just enzymes that break down the peptide bonds between proteins, things like histamines or lipids. And these mediators are the way pathogens encourage the spread of themselves. So these are what cause you to be sneezing and sniffly when you're sick, vomiting, have diarrhea. All of those are due to these inflammatory mediators, and they also help make you miserable. So this really begs the question, how does the rhinovirus cause asthma exacerbations? I mean, my talk is not about how lipids make you sad, it's how the rhinovirus is causing asthma. So, like it says, bullet point right on top, 50% of all asthma exacerbations are actually caused by the rhinovirus. So what happens is, first, asthmatics acquire the rhinovirus. Step one, check. This triggers an inflammatory mediator production, and as a result, you have an asthma attack. Seems easy when it's on this really tiny slide. Really, you can have variation in the intensity of the asthma attack. It can be more mild, it can be more severe. There are several different kinds of inflammatory mediators that can be produced as a result, and the type of rhinovirus you can get also is varied. So, seems simple, is actually very complicated. So, rhinovirus, yay! The next big, major, noticeable word on my title. The rhinovirus is a positive single-stranded RNA virus and is part of the Qpcr family. Other members of this family include things like polio, foot and mouth disease, and hepatitis. So, quite the formidable, fun-named family. The rhinovirus is, as I have previously mentioned, responsible for the majority of common colds and a lot of asthma exacerbations. Over there you've got a nice little rhinovirus as drawn by Microsoft Paint. And then in the two corners you can see a more scientific depiction. 1A is down in that corner and 16 is up there. They look pretty similar. They are fairly similar. So sorry, I'm glad you guys are laughing though. It makes it a lot easier. So because the rhinovirus is a positive single-stranded RNA, it makes it very easy to replicate in your epithelial cells because the genome is read as mRNA. So if your cells are copying, part of the transcription process is copying over mRNA. So because it's a single-stranded positive molecule. So when the virus injects its genomic information into the cell, it's read as mRNA and immediately transcribed into proteins. The virus also carries with it some viral proteins that help modify it and put it into the viral genome. Those then get packaged and sent out to wreak havoc on the rest of your immune cells. So this brings us to our first lab hypothesis. We believe that because macrophages are key in releasing the inflammatory mediators during a rhinovirus infection. So why is this? Well, they are the most prominent immune cell in your airway, and it's important because they cannot be productively infected by rhinoviruses. So they're not turning into little virus pumping factories like other immune cells may be. The macrophage's main job in the immune system is to phagocytose any cellular debris, so any worn out dead cells or pathogens, as Shannon pointed out in her last talk. And they're also in charge of stimulating other immune cells to come to the infected area. So all of you with high school bio, if it was 20 years ago or 5 years ago or last year, I don't know, immune cells like the T cells, B cells, and other lymphocytes, they're going to come to that area due to the inflammatory mediators released by macrophages. So this begs the question, how do the rhinovirus encourage macrophages to release inflammatory mediators without replication? I know you're all thinking it, so were we. The answer to that is signal transduction. It's our favorite biochemistry process, yay! So what really happens, to simplify it for everyone, is some sort of substance binds a receptor on the cell. This triggers a cellular response, and some sort of biological endpoint is resulting from that. Here you see the rhinovirus come in and bind to a receptor. This activates the G protein RAC, which then goes on and triggers activation of the P38, ERK5, and NF-kappa B pathways. As those are all more signal cascades themselves. Eventually, something happens and is brought into the nucleus to affect things like transcription factors themselves, the production of genes, or in our case, in our interest, the inflammatory mediator production and release. But it gets better because rhinoviruses not only have one receptor, but they have two. Science! So, during our research with Sonia and Dr. Dave, we looked at the rhinovirus 16 and 1A, and both of those bind to different receptors. Rhinovirus 16 is part of the major group. It's part of the 90% of virus strains, or rhinovirus strains, that bind to the ICAM1 receptor. ICAM1 is basically just in charge of cellular adhesion and stickiness, making sure you're one solid molecule and not a bunch of thousands, billions of cells just floating in one area. And the rhinovirus 1A is part of the minor group, so 10% that bind to something other than ICAM1. In the case of rhinovirus 1A, it's binding to the LDL receptor, which is in charge of bringing in cholesterol because it's the low-density lipoprotein receptor. Duh. So because 16 and 1A are binding to these two different receptors, we thought that there might be a different signaling cascade occurring between them. And this would eventually mean that if there are just differences occurring in these two signaling cascades, there's no real general response to viral stimulation. So this brings us to our second lab hypothesis. I'm going to really hammer this point home for the next 20 minutes or so. Virus 16 and 1A, they bind to different receptors, so there's going to be some sort of differential inflammatory mediator production occurring between those. So our experimental design, first we took blood from willing donors. Dave, myself, Kasonia, Dan, other friends that were willing to let me stab them with a needle. We centrifuged that down and isolated the macrophages from their blood. We exposed those macrophages to rhinovirus 16 or 1A for a varying amount of time, and we finally sent a sample in to get RNA sequenced. So RNA sequencing, for those of you who do not do science, is part of a next-generation sequencing technique. It sounds like it comes straight from Star Trek. It does not. We're in the next generation now. Surprise. What it does is you send in a sample of cDNA, and it gives you results of the RNA content in that sample. So unlike your DNA, where you have your 4 different bases and they stay relatively the same throughout the entire genome, your RNA varies. The genes in your RNA vary depending on what kind of environment they're in. So we wanted to expose them to an environment with macrophages and see how your RNA would change. And this is what we got sent back to us when we sent in a sample for the RNA-seq. What you have here is on the x-axis, you have the number of genes in a control, and on the y, you have the number of gene reads in rhinovirus 16 on the left and for rhinovirus 1A on the right. And if nothing was happening, if we put the macrophages in and they were like, it's chill, bro, nothing would have happened. We would have had a 1-to-1 ratio, which would have been a nice diagonal line from the bottom right to the bottom left to the top right. But instead, obviously, we don't see this. We see these cool things that look like jet propulsion things. So what happens is each one of these little red dots represents one gene, and anything above or below that line is going to be up or down regulated respectively. So basically the main takeaway from this is that there are several genes that are being differentially regulated once they're being exposed to macrophages. And rhinovirus 16 causes about 300 genes to be differentially regulated, but 1A only causes about 70. So that's exciting in science terms. However, we only sent in one sample to be RNA sequenced, and it was Dave's blood. So anybody who's taken an 8 a.m. lab with Dave knows that he doesn't really respond to things like normal people. We wanted to actually confirm that genes were being differentially regulated, and it just wasn't Dave being hyper at the moment. So along with those heat maps, we got an Excel data sheet that had 25,000 genes on them. And Dave basically said, yeah, go pick some out that look cool. And so you and I were like, what? What we did is we basically selected a bunch at random and then looked up what their function was. And if they seemed like they could be related at all to asthma or rhinovirus in any way, we decided it might be cool to actually check to see how they were regulated, upregulated and downregulated. So we came up with this list, this motley crew of genes test. I'm not going to explain them all in detail because that might take me the next half hour. But it includes several cytokines, so things that are important for cell signaling, IL-1RN, which is an interleukin negative reinforcer. So interleukins are what give you a fever when you get sick. So we thought if viruses are causing that to be upregulated, the negative reinforcer, that's going to stop your fever, and your viruses can continue to replicate happily in your body. Let's see, BERK3 is an apoptosis inhibitor, so it's going to stop the cells from committing suicide once they realize they're infected with viruses. And then we've got some kinases on the right, and ZAS3/HIV is pretty cool. It was completely unknown. There was no data on it anywhere. So we're like, hey, let's check it out and see what happens. And of course, MMP12, which is the topic of my talk, so obviously I'm going to talk more about that later. So we really, we decided to test those by looking at their protein and RNA levels by doing some Qpcr to validate that Dave's just not crazy and that all genes are going to be up or down regulated when you infect macrophages with rhinovirus. So our experimental design was to run time courses by infecting macrophages for between 15 and 90 minutes, and then to either kill the cells and take them to run SDS page westerns and Qpcr with, to check phosphorylation status of the pathway we're looking at, protein levels, and gene expression, or to collect the supernatant media and run ELISAs to check on the protein production that was occurring in that environment that the macrophages were in. So we started off by looking at some ELISAs. ELISA just stands for Enzyme-Linked Immunosorbent Assay. And what it does is you use antibodies and color-changing reagents to identify the presence or absence of cytokines in your samples. So the three that we focused on for this experiment was the IL-1RN, CCL17, and 18 cytokines. So this is how an ELISA works. You have a 96-well plate and you coat the entire thing with a capture antibody. So say we were looking for IL-1RN presence, we would use the IL-1RN capture antibody. Next, we would add our samples, and if the IL-1RN antigen was present, it would attach to that capture antibody. The next step is to add a detection antibody. It'll detect your antigen in the sample, and it'll kind of sandwich your sample in between the two antibodies. Next, you add a horseradish peroxidase, which just kind of chills on top of your sandwich. And finally, lastly, you add is TMB, which allows the horseradish to change color. Stop laughing at me, Mike. So if you do have anything present, in this case, IL-1RN, everything would kind of build on each other, and eventually when you add the TMB, it's going to change to yellow. So when we were running ELISAs, we were looking at obviously cytokine production and how the three different pathways would affect how, like, whether they were upregulated and downregulated respectively, and then also between three separate pathways. So that was pretty intense. And to do that, we used different inhibitors as well to see how one would affect more than the other. Yeah, words are not going so well right now. But we used the SB inhibitor to stop the P38 signal transduction pathway from occurring. P38 is involved with stress kinases, so if you get stressed in any way, your cells get stressed, you could get stabbed, you could be trying to run your senior presentation and have your boyfriend laugh at you. Anything that's going to cause your stress levels to go up, you wouldn't think of the word now. Jeez. You would expect to see your P38 activated and upregulated. We don't really know what ERK5 does, but we thought it would be worth inhibiting as well, so we did it because science. And we also focused more heavily in our research on the NF-κB pathway, so I'll talk more about that later, but we didn't really do anything to inhibit it. And so when we did do our ELISAs and we looked at the IL-1RN production, we saw, yay, our first hypothesis is true. We see some differential cytokine production between rhinoviruses that were given to macrophages that were 1A versus 16. Compared to the control, 1A is upregulated to about 1900 picograms per milliliter, and 16 is sent way up above 2100 picograms per milliliter. So these first three are the main focus. We're just going to hammer home that 16 and 1A are different. However, X, obviously, I just mentioned, is inhibiting the P38 pathway, and SB is inhibiting ERK5, and you can see that there's always a different response occurring between 16-1A and just the control. So to bring it back to MMP12, our hypothesis was that it's going to be differentially regulated by rhinovirus 16 or 1A exposure. This literally sounds like I'm repeating myself, but I'm going to hit that point home. It's going to be the one thing you remember. So MMP12 is it's a protein in the matrix metallopeptidase, or MMP family, and it's involved with the breakdown of extracellular matrix, which is really just the tissue that provides structural support to your cells. And why is it important to asthma? We're finally getting to my topic slide now. Well, first, when you have your macrophages and they come across some sort of pathogen, they're going to present an antigen, and that's going to cause your immune system to respond. So in the case of asthmatics, it's responding by producing extracellular matrix, and this is what causes your airways to thicken, because collagen is the main component of extracellular matrix. So the MMP12 gene, if it's going to break down that extracellular matrix, it's going to help reopen your airways. So that's exciting if you have asthma. So once again, our experimental design, we're going to try to look at the phosphorylation status of the pathways, protein levels, gene expression, and cytokine production. So to start off with time courses, because I've kind of mentioned them, but haven't really explained them in depth. What you do is you take cultured cells and then you treat them with viruses. So in our experiment, we used 10 viruses per one macrophage cell. Cool, that's on a timer. So then at specific time intervals, you kill the cells. It's great. So we did ours on 15-minute intervals between 90 and 15 minutes. And then we would either collect the cell lysate or media, as I mentioned earlier. And then we ran Qpcr to kind of look at the gene expression. Qpcr is mainly in charge of amplifying and quantifying the target DNA sequence. So you take a tiny little DNA sequence and you force it to go through a lot of temperature change with some primers, and that multiplies the amount of DNA samples you have, and that lets you know how much you had in your sample to begin with. So you start off by heating it way, way up to denature that double helix. You add primers and cool it down to allow them to attach on, and then you heat it up to about 68 to 72 degrees for prime polymerization. Then you've got like the split DNA, each has now 1. So it goes from 1 to 2, 2 to 4, etc. And then after you're running Qpcr, you get this lovely little plot here on the right. It's just showing the amplification of DNA through the number of cycles. So if you're looking at this as a baseline, it's going to compare how long it takes each gene to hit that same level. So the ones closer to the left are going faster. It takes only about 12 or 14 of these cycles to hit X level of gene product. And then these ones on the right take longer, about 34 cycles to hit that same level. So you know that the ones on the left had more genes to begin with than the ones on the right. And once again, we ran Qpcr with inhibitors just to see how that would affect the transcription of genes, how these 3 pathways would eventually get into the nucleus, and how they each respectively would regulate gene transcription. And this was our MMP12 Qpcr data. If you look on the top, compared to the control, 1A had a fold difference about 0.4, while 16 had a fold difference of about 10.4. So there's 10.4 times more MMP12 gene product being made in cells that were exposed to 16 compared to the control of nothing. And then these boxes each show the other 2 pathways where we were inhibiting X or we were using the SB inhibitor, and how that relatively changed the amount of gene products being made. Generally, the trend is 16 is upregulating much more than 1A is across the board. So really, we ask the question, what's really regulating this? Why is this happening? And again, the answer is signal transduction. I didn't want to explain that slide. Basically, I'm moving on to the other half of our experimental design now. We looked at ELISAs, we briefly looked at Qpcr, now we're going to talk about SDS and Westerns. So here's SDS page. What it does is it separates proteins that you have in your sample based on the size of them. So you apply a negative charge to the top where you load your samples in, and it causes them to run down, the proteins to run down your gel. The larger ones get trapped closer to the top, while the smaller ones make it down further, and then they get trapped towards the bottom. And from there, you go and you do a Western blot, and this transfers your proteins from the gel that you just ran onto a PDF membrane. I feel like I'm taking an eye exam. So basically, that just allows you to actually image your proteins. So if this is what your SDS page gel looked like, this would be 2 different wells with different proteins in them. You would transfer it onto a membrane, and then you would add a target antibody for whatever protein you're specifically looking for. It attaches to that protein if it's present, and then you would add a secondary antibody and wash everything else off, and everything not attached to your specific antibodies will be gone. And then you can analyze those versus based on the signal intensity, so how dark they are when you finally image them. So from that paper that Dave was talking about earlier, we figured out that there was differential signal transduction occurring and kinase activation of the P38 and ERK5 pathways. So to kind of break it down, because this is scary and very dark, we have rhinovirus 1A on the left of this line, this line, and rhinovirus 16 on the right. Right directly underneath it is the treatment that it received. So it was either control, rhinovirus 1A 15-minute exposure, rhinovirus 1A 30-minute exposure, and so on. And this is for the P38 or the ERK5 pathway. And GRB2 stands for GRAB2. It's just it's like a loading control. It just makes sure that these differences that are occurring up in the ERK5 and P38 are actually due to differences occurring within that pathway and not due to me loading half as much sample in one lane as is to the other. So it's making sure that I'm not cheating when I'm making these pictures. But generally you can see for the P38, there's more activation occurring between probably around 60 minutes is where it peaks for 1A exposure, but for 16 it's peaking around 30 minutes, and ERK5 you can see the same thing is happening where 1A exposure peaks around 60 minutes, and it peaks, actually they're all pretty dark, but it probably peaks somewhere between 15 and 30 minutes in macrophages exposed to rhinovirus 16. So yay, differential signal transduction is actually occurring. My summer was not wasted. So as I was mentioning earlier, Sonia and I mainly focused on the NF-kappa B pathway out of that huge model that I had shown. So what happens is the rhinovirus binds and causes some sort of inflammatory mediator release. That's up here. This is brought into the cell and activates the I-kappa complex here. This goes on and activates the I-kappa B alpha, NF-kappa B, P65, P60 complex, and then that splits, I-kappa B alpha splits off that complex and is just left out in the cytoplasm and is degraded, while the NF-kappa B, P65, P50, P52 gets phosphorylated several times by different kinases and is eventually brought into the cell and can affect things like transcription, cytokine production, etc. So because 16 and 1A bind to different receptors, if you hadn't heard, we expected to see some sort of differential degradation of I-kappa B alpha and differential phosphorylation of the NF-kappa B P65 occurring. You can read the title. That's what we saw. So this is one of the blots I took. I didn't include GRAB2 because I'm lazy. But what you see is this is the control. It's super light, and we see activation for macrophages exposed to 1A peaking around 60 minutes, and for the rhinovirus exposure of 16 macrophages, it peaks somewhere between like 15 and 30 minutes. So we do see differential degradation occurring. And we also saw differential phosphorylation occurring for the P65 complex. Same kind of thing happening. 1A causes peak phosphorylation occurs at about 60 minutes, while 16 peaks at about 30 minutes of exposure. So really when you combine those two slides together, you see that 1A is focusing more on degradation of the IKB alpha complex at 15 and 30 minutes because there's nothing there. It's focusing on degrading it, and then it switches over to phosphorylating P65, and more IKB alpha then builds up. And for 16, it looks like it's focusing more on phosphorylating the NF-kappa B pathway for the first 15 minutes, half hour, and then it switches over to degrading the IKB alpha complex. So we built this model, and it seemed to hold true throughout most of our research, where Human Rhinovirus 1A is going to bind to its respective receptor. Rhinovirus 16 binds to its respective receptor. It turns on the NF-kappa B pathway. There's some sort of differential signal transduction occurring between that pathway depending on which rhinovirus binds. Eventually, P65 is brought into the nucleus and is affecting the transcription of MMP12 differently depending on what's binding. So to really revisit our hypotheses, the macrophages are key in releasing inflammatory mediators during rhinovirus infection. We checked that off. That's true. We did see cytokine production when either rhinovirus 16 or 1A had bound. Our second hypothesis, that because they're binding to different receptors, we would have differential inflammatory mediator production occurring. That also checked off because there were differences in the IL-1RN graph between 16 and 1A. And finally, that MMP12 would be differentially regulated by 16 and 1A. That also checked off when we verified through Qpcr. So in conclusion, we found that there is actual differential gene expression occurring between rhinovirus 16 and 1A. It's probably the last time you'll hear it, so be thankful. We also found that there was an altered activation of the signaling pathway of NF-kappa B through rhinovirus binding, so the difference in focusing on IKB alpha versus the NF-kappa B complex first. And because there is differential expression of MMP12, we thought that there might be some sort of asthma exacerbation susceptibility depending on what kind of rhinovirus type you're infected with. So in the future, we would love to measure the MMP12 protein production. So if we could get some antibodies and measure that through SDS and Westerns, that would be awesome. We would also like to look at some more cytokines. I mean, we only looked at 3 out of the list of 370 plus. We could look at some other signaling pathways. We only knew about ERK5, P38, and NF-kappa B because of the RAC activation from previous people who had been working in Dave's lab. And we would love to look at more genes because we only picked 12 out of 25,000. So I'd just like to take a moment to acknowledge Dave for putting up with Sonia and I last summer and this year, making sure we were on the right path and helping with all of our imaging issues because we had plenty. Bryce Schuler and Michael Schreiber, for their previous work on this project, they were pretty instrumental in that ERK5 and P38 complex and the kinase activation that I talked about a little bit. Sonia, as being the best lab partner ever, running Westerns and ELISAs and keeping me sane all last summer. And of course, Sally and Dan in the stock room for making sure we had enough gels and reagents to actually run our experiments, because we ran through a lot. There's some references. And I think any questions, including what this is.
No.
In the 25,000 possible hits that you had, did you notice any other MMPs?
There actually were other MMPs. It's a pretty large family, but for whatever reason, I I mean, I haven't looked at it since literally last June after I selected the genes, but I know there were other MMPs listed on there. How high up they were in their p-value probably ranged a little bit. For some reason, I really want to say MMP 35 was on that list, but I could literally be making that number up. But yeah, I definitely remember seeing other ones.
So then why did you pick the MMP 12 out of those?
Specifically because it was breaking down the extracellular matrix. That's why I selected that one. Other questions? Yeah. This was a fort/castle that Custodian and I built out of empty loading tip boxes. That's how many gels we ran. That table's got like 3 feet worth of empty boxes on it. Do a lot. Lots of science.
Other questions?
Why are you so awesome?
I was born with it. You have to thank my parents for that. Yeah.
Is there any, at this stage, is there any practical applications for relieving asthma symptoms from this research?
Not at the moment. If you remember that slide I had in the beginning, we were saying that if there were differences, that there was no like generic asthma like symptoms that you would get, so it would be very difficult to treat it. I think if you took this research super far down the road, it could lead to genetic treatments for asthma, which would be kind of cool, knowing like how your genes would respond to the specific rhinovirus infections that you would get, and knowing like what steps to take to aid in that would be pretty cool.
Let's thank our speaker again.